In the auditorium is Dr. Waldemar Debinski. In my spare time, I studied brain tumor abstracts, and there is no way that if you did the same, you wouldn't come across this name. He's involved in numerous trials across the country, and his drugs have been invented and being used by patients today. Besides his scientific work, in which he holds 13 patents, Dr. Debinski is the Director of Brain Tumor Center of Excellence at Wake Forest University School of Medicine. He is the head of the section of brain tumor biology in the Department of Neurosurgery and professor in the departments of neurosurgery, radiation oncology, and cancer biology. Dr. Debinski invented the drug currently in use at many cancer centers for the precise trial. It is called IL-13-PE38. This trial is showing some very good results. Please welcome Dr. Waldemar Debinski and his friend who will help him with the slides. He could not come today. His plane was canceled. Thank you.
Thank you, Dennis, for this nice introduction. I really apologize because it never happened to me that 2 flights were canceled in a row. So that's why I cannot be with you. Do you hear me well?
Can everybody hear in the back?
Sheila, can you hear the doctor talking? Not yet. Say something again, please.
Waldemar, go ahead and say something.
So I'll be talking and just you can adjust. I never done this before to talk to the audience I cannot see. And I have a pretty rich demonstration that Phyllis will try to help me to show you today. So please bear with us. But I appreciate the opportunity to talk to you, and I also would like to thank Alma for inviting me for this event. So let me start, and when you hear next slide, it's not a part of the presentation, just a command to change the slide. Next slide. I think that what we need to realize that during the last, I don't know how many years, maybe 50 or so, there has been a tremendous, increase in the information obtained through scientific research, and this information was translated into a new scientific knowledge. But what is quite unsatisfactory is that this knowledge was translated into the much fewer medical applications that we would like to see. And I think I don't have to mention that, especially in the area of brain tumors, and especially in the area of malignant brain tumors, we would like to see a much bigger progress. So this is a tremendous challenge for us, and also a tremendous stimulation to do the best possible job. Next slide. My lab is interested primarily in finding pharmaceutically tractable targets in brain tumors. What does this mean? Well, not looking just for the targets to study them or explain their scientific significance, but we would like to identify those targets which can be used for making medicine. And it's a very different story, and it's so much more difficult to do that than just regular research. And I think the best testimony to that we don't have that many new anti-cancer drugs on the horizon which appear for the past 20 years or so. Next. So this is like a schematic illustration what we would like to see or what we would like to achieve. Next. There's like having a normal cell, normal central nervous system or normal brain cell. And we have a glioma cell, and they are certainly different, but the difficulty is to find what is the difference, and it is even more difficult to find what will be the difference between the two that might be useful for making a drug or design a preventive measure on something of this sort. And the line, yellow line, represents that sometimes, even if the difference between brain tumor and normal cell is not a very big one, then we can still enlarge this difference by studying the pathobiology of tumors and being lucky enough to identify systems that would allow us to do that. Next slide. So as you know, especially for treatment of gliomas, what is available right now next is surgery, next is radiation therapy, next is chemotherapy, and what we put a lot of faith into are the experimental therapies. Next slide, and next slide. And among those experimental therapies, still, do you follow me?
I'm with you, Waldemar.
Yeah. Among those experimental therapies, our main interest, not only but main interest, is immunotherapy. And I believe Dr. Linda Liao talked quite a bit about immunotherapy, especially active immunotherapy this morning. She has some very exciting data coming from her lab and from her clinic. And I'll be talking about a little bit different type of immunotherapy than her. Next slide. So just to remind you, the immunotherapy is divided into active form and a passive form. An active form is primarily represented by vaccine, and and passive form next is represented by the use of activated blood cells, or antibodies as such, or cytokines, which are the factors that regulate the immune responses. Next slide. And I'll be talking about passive immunotherapy. And it sounds like passive immunotherapy that you might not need to do much about it. Just to the contrary. Passive immunotherapy takes its terms from the fact that we don't ask the body to do something against the tumors. We provide the organism with something that we feel will attack the tumors. But it's actually even much more laborious than the active immunotherapy approach. Next slide. And still maybe you can push 3 times and go to the next slide because it will be difficult to say that without being there. So this is a slide about the cancer-specific immunotherapy, and up front I would like to emphasize that there will be something which is called a cytotoxin-mediated cell death next, and next, and next, and next, which is composed of ligands, and next, and a bacterial toxin. And next, you will see the molecules like that will be attacking specifically brain tumor cells and causing those cells death. Next, and next, and then next. Antibody-mediated cell death is also something of potential clinical usefulness. Next, and this again based on the recognition of something which is specifically expressed on brain tumor cells by antibodies, or similar targets or same targets can be also recognized next and next by T cells, and next those T cells will come to cells, recognize them specifically, and cause their death. Next slide. What we've been working on is what I call passive immunotherapy using bacterial toxin containing recombinant cytotoxin. It's complicated, but the point is that we use 2 compounds which are composed of 2 components, main components. One is the bacterial toxin, the other one is the ligand, and all of those recombinant proteins mean that we ask actually bacteria themselves to produce those reagents for us. Next slide. And just to illustrate how does this cytotoxin look like, especially recombinant ones. Next. They're composed of a target ligand, next, and an effector. And the target ligand may be a protein in the form of a growth factor, a cytokine, or monoclonal antibody. And next, a bacterial toxin, we usually use Pseudomonas exotoxin A or the stevia toxin or ricin for a reason I don't have the time to explain why, but these are the three most commonly used for this approach. When combined, next, those targeted ligand and an effector, next, produce something which it's a complicated name, but I like it. It's a fusion targeted chimera cytotoxic protein, next. And this would look like that, and next, one may even suspect it might be a silver bullet. Certainly we would wish it would be one. Next. And how does it work? This targeted protein recognizes next a target which is present on cancer cells. It reaches this target, and it causes disappearance of especially cancer cells. That's how we would like to see it in the clinic. Next slide. Still, is it going okay?
Yeah, we're okay.
Yeah. So one would ask why to use this particular recombinant cytotoxin for the treatment of brain tumors. And I believe there's a list of important reasons to do so. Next, one is that those compounds are really killing cancer cells. They don't make them growing slower or changing the phenotype or morphology for a while or make them dormant. They plainly kill them. So it's a matter of elimination. Next. They are active under low oxygenation conditions, and I think for many of you it is well known that many brain tumors show signs of hypoxic regions, and it is important from the therapeutic standpoint. Next. Those reagents are active independently of programmed cell death. So if cell is, for example, a resistant cancer cell, is resistant to apoptosis, it doesn't matter because those reagents, those factors, those compounds, they don't rely on this mechanism at all. Next, what's important, recombinant protein, recombinant cytotoxin, can be and they have been administered locally, directly to the tumor, and through methods developed at the NINDS by Dr. Altfield, which is called convention enhanced delivery, CED. And this overcome difficulty in distributing the drug through the blood-brain barrier to tumors to the central nervous system. So it is very useful. It is sort of a drawback, but at the same time, it has been done now in thousands of patients, and I think that's a very valuable method of drug delivery. Next. The recombinant cytotoxin work independent of the role the target may play in tumor pathogenesis. So the target doesn't have to be like an oncogene or something like that for those compounds to be active, which is also very important. And next, one of the most important features is they do not produce readily resistance. The cells which are responsive and were responsive to the drug, and even if there is a rebound in tumor, it is not that the tumor is still responsive and susceptible to recombinant cytotoxin. So there's an important clinical feature. Next slide. And with clinical use of cytotoxin, there are certainly important issues to be considered. And some of them are listed on this slide. Next. Such a specificity of the target is number one on my list. Next. Immune response to chimera fusion proteins. They are usually foreign proteins, and they will produce immune responses. Next. They activate biological system, because if we use something like a cytokine or a growth factor, it's a naturally occurring compound, and they have their own specific targets, and they usually induce some consequences in biology. Next. There's a non-specific toxicity of the cytotoxins, such as to the liver, or they can damage endothelium, but it is dependent on the dosage of the compound. And next, and they cannot cross the brain barrier, and that is why we need the local deliveries of the drug. However, next slide. The most important issues when those reagents are used in the treatment of patients with brain tumors are the specificity of the target. And next, activation of biological system by the cytokines or growth factors. And you will find from my presentation later on how we address those issues. Next slide. So, but what would be a target in for example, malignant glioma? Next slide. When I entered the field of neuro-oncology, it was more than a decade ago. It was known that the receptors, the proteins which are present of plasma membrane of cancer cells, for cytokines, for the growth factor, of mitogen, are present in GBM next. And this was found for the epidermal growth factor next, insulin-like growth factors, several of them next, some of the interleukins such as IL-6 and IL-4, next for fibroblast growth factor, next and also for transferrin. Next, but the bottom line is that none of those receptors is specific to brain tumors, with the small exception of one of the mutated forms of EGFR. V3 is a mutated form of EGFR receptor, which is present in a minority of patients and very, very heterogeneously. Next slide. So also, I established a list of when to expect eventually that the therapies might be effective using the recombinant cytotoxin next. Especially what was the most important is the target. There's the target for the cytotoxin. And next, it must be overexpressed in tumors versus normal tissue. Next, this target must be present in a vast majority of patients with brain tumors, because then it makes really sense to design therapies and go ahead with them into the clinic. Next, that the target must be present in a vast majority of cancer cells within the tumor, because then the issue of tumor heterogeneity is also addressed by in using such a target. And next, preferentially, such a target should serve development of at least two types of immunotherapy. And I'll mention about a little bit later during my talk. Next slide. Is it going well, Lisa?
We're doing great.
Good.
1 second.
Okay, I should have asked.
Sorry about that. All right, we're set.
Okay, so one of the targets which is close to be optimal and which fulfills most of the criteria to be a good target for recombinant cytotoxin is a receptor for interleukin-13. But one needs to remember that there are two types of IL-13 receptor. One which is present in normal tissue next, and it is a receptor which is a physiological one, which serves a lot of good purposes under normal circumstances. And it is shared with IL-4, and also it's a complex receptor because it contains two subunits. Two subunits are needed to form this receptor. However, what we found is that there's another receptor for IL-13, which is we call it tumor-associated receptor, which is very different from the one which is in normal tissue. And also, what's different is that it doesn't bind IL-4 at all. And now we know that this restrictive IL-13 receptor is a molecular entity called IL-13 receptor alpha 2. Next slide. And just to ask what bacterial toxin would be suitable for immunotherapy using some recombinant cytotoxin. Next slide. There are two that I've been working mostly, and one, pseudomonas-toxin, simply because of my first PhD training at the NCI a while ago. I don't even want to say how many years ago. And this pseudomonas-toxin is secreted under normal circumstances or pathological circumstances by Pseudomonas aeruginosa bacteria which are very vicious and dangerous in a hospital. And one of the reasons why they are so dangerous is because they have really powerful weapons to use. Next. And one of those weapons is Pseudomonas exotoxin A. And next. And next. And 5 times, 6 times next, please. Because then you can see this bacterial toxin is composed like several units, and one unit is responsible for the binding to the receptor. The other subunit is responsible how to sneak this bacterial toxin into the cell. And the last unit, which is called E, is an enzymatic ADP ribosylation domain. And this is the domain which really makes a eukaryotic cell kill. Some say that only one molecule of such a toxin is enough to kill a eukaryotic cell. I think more is required, but not that many more. Next. And there's what's called P66 because of its size. Next. What genetic engineering next allowed us to do is to model the Pseudomonas exotoxin in a way that, for example, it doesn't have its receptor binding domain. So under those circumstances, and that was what can be called P38, because it's a smaller compound, it's a truncated form of Pseudomonas exotoxin. This truncated form is not active on its own. Actually, it's impotent and it doesn't do much harm to eukaryotic cells, including cancer cells. However, one can introduce something else in place of the regular PE binding domain, such as IL-13, and make a targeted cytotoxin, which I will show you a little bit later on. Next slide. Are we on the next slide?
Where the diphtheria toxin?
Yeah. So another toxin frequently used, although the field is not that big, I'm saying frequently it's by those who are working in this field by us, is diphtheria toxin. And this is a mirror image of Pseudomonas exotoxin, still 6 times, okay? Because and 7 maybe. Because how does it look like? Diphtheria toxin is the opposite of Pseudomonas exotoxin. It has a receptor binding domain and at the other end than PE. Otherwise, they work in a similar fashion. They kill cancer cells or other eukaryotic cells in a similar fashion. And again, next 3 times. This using genetic engineering, one can make a truncated diphtheria toxin and allow this such a protein to be redirected by another ligand that can be added to it. Next slide. How do Pseudomonas exotoxin or diphtheria toxin or the cytotoxins counterpart work on eukaryotic cells? Next. Do we have a picture?
We do.
Yeah. And if you look at the top of the panel, you can see that what is required is that for a molecule of Pseudomonas exotoxin composed of those 3 different fragments, this toxin, all the cytotoxin based on Pseudomonas exotoxin, must bind to a receptor, and once it is bound, then it is so like taken up by cells in a process which is called ligand receptor internalization. And those cystic vesicles which are formed during this process are maturing further, and during this maturation at pH, the environment changes in those vesicles that allows to a cleavage of Pseudomonas exotoxin into a form that can go out of such a vesicle, and that's what is called translocation to cytosol. And once it's a cytosol, it looks for its a specific molecular target, which is the EF2, as you can see it as on your right-hand side at the bottom of the panel. And through a specific chemical reaction, the enzyme EF2 is inactivated by bacterial toxin, and this inadvertently leads to a reversible cell death. Next slide. Next, this 4 times next, please. The first cytotoxin which has been ever used in the treatment of patients with brain tumors is an immunotoxin based on transferrin and an inactivated form of diphtheria toxin. And this is a chemical conjugate. Next. And what's this was a pioneering studies by the National Institute of Neurological Disorders group, which I would honestly say I would have courage to do anything similar to that, simply because of the relatively non-specific form of the immunotoxin, because the target for this immunotoxin is also present in a large amount in normal brain. But anyway, they're very skillful and they know this compound very well. So in their clinical trials, the clinical responses were observed. Next, the phase 2 clinical trials even were completed. And what's very interesting, and I was a little bit surprised, but still happy to see this happening next, that the phase 3 clinical trial began. And I don't know much about, I haven't heard many news about how does it go, but I think it should be finished within the next year or so, and then we'll know how a relatively non-specific cytotoxin was doing in a controlled clinical trial. Next slide. So let me talk a little bit about the interleukin-13 system, the system that we work on for the past more than 10 years now. Next slide. And let me start and remind you about IL-13 receptors, because this is the clue to the whole story. Next slide. As I mentioned, there's a normal tissue heterodimeric receptor for IL-13 that is shared with IL-4. Next slide. Sorry, not next slide. Next. And next. Which is composed of 2 completely different subunits. One which is called IL-13 receptor alpha 1. Next. And the other one is IL-4 receptor alpha. Next. These 2 are needed to produce an effective binding, next, of IL-13 to this receptor. That's what happens in physiology. And next, IL-4 is also a competitor for this receptor. So they're sort of like siblings. They're similar, and they interact with the same receptor. Next slide. Actually, it's the next. And next. And that is why this situation can be reversed. So IL-4 can bind to this receptor, next, and IL-13 is still a competitor around and a candidate to interact with this receptor. Next slide. However, what we discovered, and at the beginning nobody believed that is the case, is that there's a restricted tumor-associated receptor for IL-13, which is called now IL-13 receptor alpha 2, which is not shared with IL-4. Next. So this is just to remind you how does the normal tissue receptor look like, and how does it interact with IL-13 or IL-4. Next. But there's another species, another protein which is called IL-13 receptor alpha 2. Next. It's a tumor-associated receptor. Next, which binds IL-13. Next. But it doesn't bind IL-4 at all. And that's the clue to the whole story. Next slide. Next. And what we found about this glioma-associated receptor for IL-13 is that a vast majority of established GBM cell lines and explant cells overexpress binding sites for IL-13. Next. That the vast majority of GBM tissue specimens overexpress binding sites for IL-13 in situ. Next. Also, that a vast majority of IL-13 binding sites in GBM is more restricted, means that whatever binds to GBM is rather through the IL-4 independent receptor than the one which is shared with IL-4. And we also documented and we've proven that IL-13 receptor alpha-2 biochemical entity is the GBM-associated binding site for IL-13. Next slide. Next. And I have to say that this work was published, we published this work a decade ago. And I thought that we might be the first one to say that we identified the first factor that is that is present in the vast majority of patients with GBM and not in normal central nervous system. However, I have to give credit to two other centers because they identified, two centers identified two different genes, but they still don't know what kind of protein correspond to those genes, which are detected in GBM but not present in the normal central nervous system. What I can say is that restricted receptor for IL-13 as of today was the first one which can be pharmaceutically tractable and is used for translational research in brain tumors. And the good news from this whole story is that molecular denominators of GBM do exist. How to translate that to you? It is, when I entered neuro-oncology field, the dogma was nothing like it exists for GBM. So if we can identify something, it will be relevant to less than 50% of patients. And it's not correct. And we were the first ones to really show that this is not the case. And now we take, of course, for granted that a new factor, a new gene identified in GBM and not in normal brain is present in 60 to 80% of patients, or even up to 90% of patients. And that's becoming like a daily occurrence now. Next slide. Also, I would like to show the scheme which demonstrates that, like normal astroglial cells and normal brain cells, they do not have IL-13 receptor alpha-2 next, but next and next, when it may progress to glioblastoma multiforme, there's a lot of the receptor present in GBM. Next. Also, we checked for lower grade gliomas and found that in grade 2, there's not that much of this receptor. Next. But next, when it progresses to grade 3, there's more and more of this receptor. And again, when in glioblastoma multiforme, next is present in the vast majority of patients. Next. What was also interesting, next, that the most benign brain tumors, pilocytic astrocytoma, has actually 100% representation of this receptor, and we're still trying to understand why it is so. Next, but we know that very few, very rare cases of pilocytic astrocytoma, next, they may also progress to glioblastoma multiforme. Maybe there's some kind of a link between in this process. Next slide. Why don't you go and simply do that? We move to the next slide because it's just too much to show and talk about. Okay.
Okay.
I wanted to show that how we demonstrated the binding of IL-13 to brain tumors, but it's a little bit too rich of a slide and it will be difficult to explain.
Okay, we're on the next.
So next slide is entitled Comparing Autoradiography with Immunohistochemistry. And next, my lab at the very beginning used autoradiography, meaning labeled IL-13 as a ligand for detection of the receptor in GBM, simply because there was no antibodies. And again, we could work with IL-13 to do that. And in our collection of specimens, we detected IL-13 receptor in around 72% of patients with GBM next. And a quite recent study of my colleagues, which is coincidental, they are from Wake Forest. Before my coming to Wake Forest, they did the study, and they detected IL-13 receptor, specifically IL-13 receptor alpha-2, using monoclonal antibodies, so very specific antibodies to this receptor. and they found 79% of patients with GBM expressing this success. Next. So what would be the utility of the IL-13 system in the treatment of GBM? How we can take advantage of the fact that there's the pathobiology, there's such a pathobiology of a cytokine receptor in grade 4 astrocytomas. So next slide. This is about the interleukin-13-based cytotoxin containing Pseudomonas exotoxin. And a small digression here. The very first generation of IL-13-based cytotoxins containing Pseudomonas exotoxin was designed and produced in my first independent laboratory in Montreal more than 12 years ago. And it has nothing to do with brain tumors. Actually, what I wanted to achieve is to sort of make a comparison between interleukin-13-based cytotoxin and interleukin-4-based cytotoxin, simply because IL-4 cytotoxin was sort of like my last subject of investigation during my post-PhD training at the NCI, and I saw that with the cloning of a new homologue of IL-4, so that's what happened during the move to Montreal, interleukin-13, that I would like to just compare the two and see what's going on, but especially using adenocarcinoma models of cancer, peripheral tumors. I didn't have brain tumors in mind, totally, completely on my mind at that time. So next, what we did is combine a wild-type IL-13 with a truncated form of Pseudomonas exotoxin, which we term IL-13P38 QQR. Those QQR stand for some very fancy genetic modification that we made to the toxin for some other reason. And this is the first generation of the IL-13-based cytotoxin. However, I started my laboratory of tumor biology and direct and brain tumor research at Penn State College of Medicine 12 years ago, and I moved specifically into the area of neuro-oncology and joined the Department of Neurosurgery because I thought application of the cytotoxin first in patients with brain tumors. And I think it was very, very coincidental because with the move we also continued to work on IL-13, and to make the long story short, my lab was empty at Penn State, but I had a tissue culture incubator and a cytotoxin in my pocket, and I had to train my technician. And what I did is I gave IL-13P38 QQR to her as a negative control, and for checking of some other cytotoxic activity on brain tumor cells. And she started to come back with amazing results, and they were so amazing. I look at them, and I thought that this is really a lost case. I won't be able to teach this girl anything in medical research techniques. But it appeared that after the third time, I did this experiment, and I got exactly the same what she was getting, and the result was such. IL-13 P38 QQR was killing brain tumor cells at such high potency that I never seen any cytotoxin in any model of cancer that would be so effective. So we started to understand that we are facing with something really extraordinary. And in investigating further, we identified that this restricted receptor for IL-13 is truly present in the vast majority of cases with GBM. We also started to work more specifically on IL-13 as a ligand. And on this picture, you see that the IL-13 has been modified because we learned a little bit about the structure-function relationship of IL-13 and started to make the forms of IL-13 in a way that would recognize even more specifically brain tumors and not attacking normal tissues. And next, This represents a second generation of the cytotoxin, IL-13, E13K, PE4E. I don't have the time to explain everything about why it is called like that. But this cytotoxin is showing better efficacy and better specificity than the first generation. However, as a clinician of physician scientist, it's sort of like a dream coming true that something that we were working in the lab may be used in the clinic. That's what we're working for. And truly, IL-13 P32QR is being developed by a pharmaceutical company. It obtained orphan drug and fast track designation from the FDA. And phase 3 clinical trial began in 2004, and it's very rare, as you are well aware, to have a candidate drug which is making in brain tumors from phase 1 to phase 3 clinical trials. And I certainly would love to hear more what Denis said, that it is showing well. I think that phase 1, phase 2 was very, very successful, but let's see how good is this drug. The second generation of the IL-13 based cytotoxin will be in the clinic by the end of 2006. Next slide. And next.
Dr. Debinski, can I interrupt you for a second? I think we only have 3 or 4 more minutes. Is there any, should we find some high notes to hit, or?
Okay, I thought we started 10 minutes later, but it's okay. Let's go for another 5-10 minutes, and then I can take questions, okay? And in terms of recombinant IL-13 based cytotoxin, as of today, they are arguably most potent anti-glioma agents. Next. They are highly potent in vivo. Next. They demonstrate encouraging clinical responses in early phase clinical trials. Next. But the clinical optimization has just started. I think this is just the beginning of the story, and we are in our lab designed the fourth generation of the IL-13 cytotoxins and working on even on the fifth one, simply because the science is progressing. We learn much more about the target, and we learn much more about the ligand. And also we learn about the biology of tumors in a way that we can incorporate this knowledge into the design of novel generation of cytotoxins. Next slide. And I think if I have that a little time, still, can you go to escape and then move the slide to the point that I would like to at least acknowledge who took part in those investigations, okay?
Do you know what slide number that is?
I'll tell you in a second. This is slide number 52.
Okay.
So, too bad I think that I could be a little bit more efficient during the presentation on my own. I had to interject the words next slide so many times. But I would like to acknowledge all people involved in the project on IL-13 and IL-13 cytotoxin in my lab. Sonia Girard, Robin Miner, Anthony Failure, Dennis Giebel, Becky Sagal, West, Kevin Nash, Jeff Thompson, Akiba Min, Madam Kumar, and Yamping Ku. Also Dr. James Connor, who at Penn State was extremely helpful in allowing us to start up especially autoradiography studies in GBM. And from outside the lab, I have to emphasize and underline the help and collaboration of Dr. Jan Lesby from the UAB. And next slide. I would like to thank National Cancer Institute for continuous funding of our research. Thank you very much.
We have time for any questions for the doctor? Yes, Al. Can you hear that, sir?
I didn't hear that.
Can this be used on spinal GBMs?
That's a good question. I think what the question is about the electrolaryngeal disease, and I think it can. all depends on how cells express the IL-13 receptor, because the delivery into intrathecal space, I think, will work out. And this question has been asked by two oncologists at Wake Forest and who would like to eventually do the trial in patients with retinal GBM and using IL-13. Yes, I think that should be done.
Okay, thank you. Any other questions? Yes, sir.
How is it introduced into the body? How do they get it into the brain?
How is it introduced into the body?
It is introduced in a way of catheter placement through the skull directly to the tumor site.
Did you hear it?
Thank you.
Yes, ma'am.